Hey everybody, we are here for our first video on blood vessels and the vascular system. So of course, there are three basic blood vessel types. In alphabetical order here, arteries, capillaries, and veins. This is also our order of blood flow. We flow from arteries to capillaries to veins. And of course, if we have our flow, so we're flowing from arteries to capillaries to the veins. And of course, if that is our flow order, that is also our order of pressure. Okay, arteries are going to have the highest pressure because they are closest to the heart, and veins are going to have the lowest pressure because they are farthest from the heart. All right, so three basic blood vessel types. Let's talk about each of them in turn. So arteries. Arteries are defined by the fact that they go away from the heart. So all arteries take blood away from the heart. As they do so, they branch and diverge. Big artery branches into smaller ones, and I get smaller ones and smaller ones and smaller ones. Now, arteries are typically oxygenated. So most arteries are going to be red on your models and your specimens and your pictures. Like these coronary arteries that I see here on this heart are red because they're oxygenated. Of course, there are always exceptions to rules. And our exceptional, our unique arteries include this guy right here. This, of course, is the pulmonary trunk. So the pulmonary trunk, as well as the pulmonary arteries, they are arteries because, again, they're going away from the heart, but they're weird because they're not oxygenated. Pulmonary arteries are exceptional. Also, the umbilical arteries, which are going to the placenta, those are exceptional as well. Okay, so that is my basic facts about my arteries. Let's do veins. No, let's do capillaries. Of course, let's do capillaries. They're next in line. Capillaries are our smallest blood vessel. You probably have around 10 billion capillaries. That is huge, and that gives you a ginormous surface area, and that huge surface area is excellent for exchange. Here I have a capillary in cross section, not much bigger than a red blood cell. Here again is a capillary right here, a red blood cell inside it. So capillaries are super tiny. And let's now move on to veins. Veins, by definition, go towards the heart. So arteries went away, veins go towards the heart. As veins go towards the heart, little veins come together to make bigger veins and bigger veins and bigger veins and bigger veins until we get our super big superior and inferior vena cava. So veins are converging. Veins are not diverging. Veins are not branching, veins are converging. Now veins, like these coronary veins here, like our inferior vena cava here and our superior vena cava here, veins are typically deoxygenated. Although, of course, there are exceptions. And the exceptions are going to mirror what they were for the arteries. Our pulmonary arteries were weird arteries, so no surprise, our pulmonary veins, here's a pulmonary vein, pulmonary vein, pulmonary vein. Pulmonary veins are exceptional. They're veins because they're going towards the heart, but they're exceptional because they are oxygenated. Similarly speaking, the umbilical veins are weird in that they are oxygenated veins coming from the placenta. All right, so that's my veins. Let us now talk about the basic structure of a blood vessel. Generally, we think about a blood vessel as, it is a tube, so it has a lumen or a space within it, and it has a wall. This vascular wall is usually going to have 3 layers. And we don't use the word layers. Instead, we use the word tunica. So a tunica is a layer or a coat. And the innermost layer is called the tunica interna, also known as the tunica intima. Then the middle layer is the tunica media, which is primarily smooth muscle. And then the outer layer, and I just realized I got a mistake here. I love catching mistakes. The outer layer is called the tunica externa. Externa. It is also called the tunica adventitia. We'll talk about why that is in a moment. So the outer layer is the externa. That totally makes sense. External, right? The inner layer is the interna. That totally makes sense because that's internal. And the middle layer is the media. Media literally means middle. So, always making perfect sense in anatomy class. Love the way that works. Here I have a beautiful picture of an artery. This is a gorgeous picture right here. I'm sure you're equally enamored by it like I am. Got nice lumen here. Look at those blood cells, red blood cells. I see what looks like a neutrophil right there. Platelet. Is that a platelet? Perhaps, or a lymphocyte. Hard to tell. But yeah, there's probably a lymphocyte right there. Awesome blood cells in the lumen. And I can totally see my three layers. This is going to be my tunica interna right here. That's my tunica interna. Let's switch colors. I'm not loving this green. Then I got my tunica media. Tunica media is right here. And then out here I got my tunica externa. So pretty cool picture. Definitely see those layers very nicely. And let's talk about those layers a little more. The tunica interna or intima. Intima is intimate with the blood. This is the lining. It is going to contain the simple squamous epithelium, which is called endothelium. Like endo means within, thelium kind of means like the room. So we're within the room, we're lining the room. This layer, this intima or interna, is continuous with the lining of the heart, which of course was the endocardium. Now, underneath that endothelium, there is a little bit of connective tissue, a little loose connective tissue supporting it. And by the way, this is the only layer that is found in capillaries. So remember, capillaries are, the job of capillaries is exchange. And so it makes sense that it's just going to have one layer, easy for things to get through. All right. Let's jump to the tunica media. The tunica media is mostly muscle. So here's our tunica media here. Media is mostly muscle. I like the alliteration on that, of the M's going together. Media is mostly muscle. And it's smooth muscle, which is involuntary, so we don't control it. There are also elastic fibers. Remember, elasticity does not mean stretchy, so gum is not that elastic. Gum is stretchy. A rubber band is elastic, meaning that it stretches and recoils. And this is really important for arteries, the ability to stretch and recoil. And so no surprise, the most prominent layer in arteries is this tunica media. The big tunica media in an artery also makes the artery strong, which helps it deal with the high pressure of the blood in there. Because we know arteries have the highest pressure of all three of our blood vessel types. All right, so tunica media is the middle layer. The outer layer, also called the adventitia, is our tunica externa. And I can see it. Let's draw a little arrow on it. I can see it right here. Bum, bum, bum. There's my adventitia. And this adventitia or externa, I like both names. I like the name externa because it's on the outside. I also like the name adventitia because basically the word Adventitia means it's something like from the outside, okay? It's like not a crucial part of it. And the adventitia is actually often shared between neighboring blood vessels. Like if I had an artery and a vein right here, the artery and vein would have an adventitia around, and the adventitia would actually be shared. So that word has to do with that like shared nature of it. Anyhow, what does the adventitia do? The adventitia anchors. Now, veins generally have a big adventitia, mostly because veins have a small interna. The interna is always small, so a small interna and a small media. So kind of like by default then the adventitia or externa is the biggest. Anyhow, this is our anchoring layer. Anchors a blood vessel to surrounding tissues, other blood vessels, nerves, organs, whatever we got. So those are my three basic layers. I want you to look at these two. I have an artery and a vein here. First of all, can you identify the artery and the vein? Second of all, can you point out the three layers in each of these guys? So go ahead and do that. Let me know what you get. Be the first person to come up to me and answer one of these questions I give you in the video. Okay, let's talk about some basic types of arteries. There are going to be three basic types of arteries. Good things come in threes. And the first type is the elastic arteries. No surprise, these arteries are going to have a lot of elastic fibers, which lets them stretch and recoil. The ability to stretch and recoil is crucial for vessels that are near to the heart. And of course, what vessel is nearest to the heart but the aorta? So the aorta and its major branches are going to be elastic arteries. Do I have a good picture coming up? No, I don't, do I? Boo, I didn't put a good picture in here. I should have. Sometimes I do these videos and I'm like, man, I should have had a better picture. It's all right. Anyway, the aorta and its major branches are elastic arteries. So I see some of the major branches on this picture here. Let me draw a heart, anatomically correct heart. There it is. Boom, 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 boom. Let me draw an aorta coming up and coming down right there. I know the aorta has three branches off of its arch. So the aorta is an elastic artery, and all three of these branches are elastic arteries. The brachiocephalic, that first one there, is an elastic artery. The left common carotid, the middle branch off the arch, is an elastic artery. And the left subclavian, that third branch, is an elastic artery. And then as the brachiocephalic comes up, it splits fairly quickly to give me a right common carotid and a right subclavian, and those are also elastic arteries. If we were to follow that aorta down, so ascending aorta, arch of the aorta, and then we follow it down, in the belly button region, the aorta splits, and the two guys that result from the split are known as the common iliac arteries. That would be a right common iliac artery down there. So those major branches are elastic arteries, plus my aorta itself is an elastic artery. And one of the things these guys do is they act as auxiliary, so helper pumps. Well, how do they act as pumps? Well, when the ventricle contracts and blood is ejected into those elastic arteries, the elastic arteries stretch. And then while the ventricle is relaxing, the elastic arteries are recoiling, because remember, elasticity is stretch plus recoil, and the recoil helps force blood onward. And that's moving blood, so helping to move, helping to pump blood. All right, my next type of artery are my muscular arteries. Most of the arteries I'm going to name, so if I, like remove my elastic arteries, the remaining arteries that I've got, those are generally muscular arteries. And muscular arteries, like their name implies, has a big tunica media with a lot of muscle. This is a cross-section of a muscular artery down here. Check out the lumen. We see the intima or the interna. And then we also see the tunica media. Okay, and the tunica adventitia outside. And the media is the biggest here. Big media is good for dealing with high pressure. I got some muscular arteries here. This is a hand, so we got a brachial artery up top here. Got a radial artery on the thumb side and an ulnar artery on the pinky side. Those guys are all muscular arteries. In fact, pretty much any artery that is not a elastic one is going to be a muscular artery. So if I'm looking at this picture here, if I take away the aorta and the common iliacs, the brachiocephalic, the common carotids, and the subclavians, everything else on this pic is going to be a muscular artery. All right, my smallest artery are my arterioles. And my arterioles are so small that sometimes they don't always get an externa. Now, one of the key facts about the arterioles is they have a lot of neurons going to them. There are these neurons called vasomotor neurons that are going to go to or innervate arterioles. And primarily the neurons are going to the tunica media, which is the muscle. And so by having neurons that go to the media, we can cause the media, the muscle there, to contract or relax. And that is going to change the diameter. And by changing the diameter of arterioles, that allows me to regulate blood pressure and regulate blood flow. So arterioles are super important because they are my sites of regulation of pressure and flow. They are where I regulate pressure and flow because it is easy to change the diameter. I change the diameter by affecting my tunica media, which is the smooth muscle. And I do that because I have a lot of vasomotor neurons that go to that tunica media. Vaso means vessel, motor means movement or mover. So the movement is involved in that diameter change. All right, let's talk about that tunica media in the arterioles. So the smooth muscle in my tunica media has tone, and muscle tone is sort of like a halfway state of contraction. It's like you're not totally contracted, nor are you totally relaxed. And the nice thing about a semi-state of contraction is you can contract more or you can contract less. Like you can go in both directions. And the muscle tone of my media is affected by the vasomotor neurons that we mentioned a moment ago. And by the way, those neurons belong to my sympathetic nervous system, my fight or flight system. But that muscle tone is also impacted by hormones as well as metabolites, like things that are produced by working tissues. That's what we mean by a metabolite. Okay, let's look at a pic that might elucidate things a bit. So here I have an arteriole. It is in a semi-state of contraction because a vasomotor neuron is always releasing some norepinephrine. Now, so what can I do? I can do two things. I can release more norepinephrine and get that tunica media to contract more and get my vessel to constrict. Or I can release less norepinephrine from that motor neuron, get my tunica media muscle to contract less, to relax in other words, and the blood vessel dilates. And this is going to let me regulate flow. More flow goes through more blood flows through a dilated vessel than constricted vessel. This lets me regulate pressure. Squeezing a vessel raises the pressure. And so this is a really important thing. All right. We are doing some great work today. 20 minutes in, but just moving along nicely here. I got 3 questions. No, I can't count, can I? I was just talking about how good we're doing today. And then I said 3 questions when obviously there are 4 questions. So do these 4 questions. If you haven't done them already, let me know what you get if you have any trouble. And let's talk about those capillaries or capillaries. I feel like British people say capillaries. What do you like better, capillaries or capillaries? Not really sure. Anyhow, capillaries, billions of them, exchange sites. I'm able to have billions because they're tiny. I'm able to be great at exchange because the wall is really thin. 1 layer of simple squamous epithelium here. Just the tunica interna. And I have capillaries almost everywhere. The only place I don't have capillaries, I don't have capillaries in epithelial tissue. I don't have very many capillaries in tendons, in ligaments, in the cornea of my eye, in the lens of my eye either. All right. Let's talk about the 3 basic kinds of capillaries. The most common type and least permeable type of capillary are these guys known as a continuous capillary. So this is a pic of a continuous capillary down here. So check out the simple squamous epithelial cell. This is one right here. Another one down here. I'm just going to outline a simple squamous epithelial cell right here. And the gray stuff on top of it is like connective tissue. But there is my simple squamous epithelial cell. Notice there are no holes in this cell that I've outlined here. I am going to super quick jump to the next slide. And you're going to see holes. And in the next slide, you're going to see more holes. So a continuous capillary has no holes. It does have spaces between cells. Those are called intercellular clefts. There are spaces between cells, but no holes in the cell itself. Now, these capillaries are super abundant in my skin, the dermis of my skin, where I'm doing a lot of heat radiation from my body to maintain my body temperature. I've got these guys abundant in my muscles. My muscles need a lot of exchange. My lungs for gas exchange. My central nervous system, my brain, and my spinal cord, because they are such hungry tissues. So those are my continuous capillaries. I've also got 2 other varieties. I have fenestrated capillaries. Fenestrated capillaries, the endothelial cells, which are the simple squamous epithelial cells, these guys have little holes or fenestrations. A fenestra is a window, so these guys have little tiny windows within them. There are also intercellular clefts between them. These guys are found in my intestines, my choroid plexuses, and my kidneys. In the kidneys, the job of these guys in the kidneys is to act as a filter. Because we know in the kidneys we filter blood to make urine. And what is a filter but something with a lot of little tiny holes in it, which is what this capillary type is. So We're going to see these fenestrated capillaries in the kidneys where they're doing filtering. They're also doing filtering in my choroid plexuses, and I find choroid plexuses in my ventricles in my brain. I got 4 ventricles in my brain. I got 4 choroid plexuses as well. And this is where I make CSF. Cerebrospinal fluid is made at my choroid plexuses. Finally, I've got them in my intestine, my small intestine, where this capillary type is super useful because I'm absorbing nutrients, and having all these holes facilitates that nutrient absorption. Okay, excellent. We got one more type of capillary. Our final capillary type are called the sinusoids, also called sinusoidal capillaries, also called discontinuous capillaries. And these guys are my most permeable, my least common. They've got big holes, so not little fenestrations, but big holes in their endothelial cells and big intercellular clefts. I find these guys in the liver, spleen, and red bone marrow. It makes sense that I find these in the red bone marrow. And here's some red bone marrow over here in the femur here. It makes sense that I find these in the red bone marrow because, of course, what happens in the red bone marrow? Hemopoiesis or hematopoiesis. That is the production of blood cells. And those blood cells need to get in the blood. How do we make it easy to get in the blood? Well, we use big holes. I've got these capillaries in my liver and spleen, and often in the liver and spleen, I'm going to find macrophages just like hanging out, sitting on one of these clefts, sitting on one of these holes, just drawing some macrophages here. They're hanging out there looking for foreign things flowing through the blood in the liver and the spleen. Of course, in the spleen, looking for old red blood cells as well. So that's my third capillary type, the sinusoids or the discontinuous capillaries. Now, most of the time, capillaries are arranged in groups called capillary beds. So I'll have an arteriole, a tiny artery, leading to many capillaries. Those are capillary beds, and the capillary beds then empty into a venule. So don't think about it being like a straight one-to-one relationship, one artery, one capillary, one vein. Often the capillaries are in beds. Okie dokie. Now, here's the thing. I got so many capillaries, it's hard to put blood in all of them all the time. So what I've got at the opening to my capillaries, so this is a capillary bed, this collection right here. And at the opening of my capillaries, I have these rings of smooth muscle. These rings of smooth muscle are called pre-capillary sphincters. And they're going to control whether or not blood goes into the capillary beds. Right now, these precapillary sphincters, the muscle is relaxed. So the muscle is relaxed in them, meaning that the precapillary sphincters are open, meaning that blood is flowing through all of these capillaries in the capillary bed. Now, I don't always want all capillaries to be flush with blood because I only have so much blood. I can't have it go everywhere all the time. So one of the things I can do in tissues that are not currently that needy is I can shut those precapillary sphincters. So in this pic, the precapillary sphincters, which again I see 5 of them, 1, 2, 3, 4, 5. In this pic, the precapillary sphincters, the muscle in them is contracted. Therefore, the precapillary sphincters are closed. So the blood is just zipping right down this pathway down the middle. The blood is not filling all the capillaries in the capillary bed. These guys are empty. And this is a good way to solve this issue of having so many capillaries that I can't fill them all at the same time. And these sphincters, whether they are open or closed, depends on tissue needs. So like, for example, if oxygen levels drop in the surrounding tissues or CO2 levels rise, that is going to cause these guys to open up. And that makes sense because if these guys open up, I get more blood flow, bringing fresh oxygen, taking away CO2. Okay. 2 questions for you to do here. If you haven't done them already, answer these questions. Let me know what you get. Got 6 fast ladies here. And if you are inspired by these ladies and you decide to increase your amount of aerobic training, your running, your bicycling, whatever you are doing, what would happen to your skeletal muscle capillary density as a result of aerobic training? Well, it'll actually increase. You will grow new capillaries in your muscles as a result of cardiovascular training. That is one of the positive adaptations that you make in response to training. It's one of the things that contributes to an increased level of fitness. All right, I think we're done with capillaries, and we are. So we're actually going to call it quits. We're over a half hour in. We're going to call it quits right here. We're going to pick up with veins on our next video. As always, hope this was valuable. Let me know if you have any questions, or just let me know if you are understanding stuff. See you later.